Right. So today, what we want to talk about is the pentose phosphate pathway. So the pentose phosphate pathway is actually like an offshoot from glycolysis, and what it does is convert glucose 6-phosphate into ribose 5-phosphate. So this is the pentose phosphate pathway, also known as the PPP. Now, I'll let you know that this actually never came up on the old MCAT. This is actually something they've added to the new MCAT. Like I said, what it does is convert glucose 6-phosphate into ribose 5-phosphate. So where have we seen glucose 6-phosphate before? Where's glucose 6-phosphate in like biochemistry?
Where does it come from?
Yeah, sure. Where does it come from?
Glucose.
So it comes from glucose, right? Glucose enters the cell. What's usually the first thing that happens to it? You learn about this in like basic bio.
Hexokinase.
Hexokinase converts it to
G6P.
Yeah, glucose 6-phosphate, right? So that's actually the product of the very first step of glycolysis. Fantastic. And what do we need ribose for?
Nucleic acids.
Fantastic, right? So we need ribose for nucleic acids.
And ATP.
Yeah, so like DNA, RNA, ATP, right? It's the ribose for ATP as well, right? So we need this ribose 5-phosphate. It's particularly important to us. And the way that biochemistry usually is bound to come up on this new MCAT is that they're going to give you the entire biochemical synthesis in the passage, right? And they basically treat it like a passage-based question where they gave you all this brand new information, and then they expect you to answer questions that deal with what happens when this enzyme is missing, or this allosteric stimulator inhibitor is like going to affect the rate-limiting enzyme, et cetera, et cetera. So what you want to know is the general setup of the triple P pathway, and that general setup is that there's an oxidative phase and there's a non-oxidative phase. Okay, so in the oxidative phase, what's the first thing that we start with?
PTH?
Well, we start with
G6P.
Yeah, we start with G6P. That's exactly it, right? So we start with G6P, and in the first step and in the third step, we form NADPH. All right? So we go from G6P, and what did we form NADPH from? What was the precursor to this?
NAD.
NADP plus. It was NADP plus, right?
Yeah.
And it goes from NADP plus to NADPH, so we hydrogenated that, which means we must have dehydrogenated what?
Glucose 6-phosphate.
Right, we dehydrogenated glucose 6-phosphate. So what's the name of the enzyme that's going to dehydrogenate glucose 6-phosphate?
Dehydrogenase.
Yeah, it's glucose 6-phosphate dehydrogenase. Fantastic. Well, when we do that, we end up with an intermediary. I don't think it's particularly important for you to know the name of the intermediary, but I'll write it out for you anyway. It's 6-phosphoglucolactonase, lactone, sorry. 6-phosphoglucolactone. And 6-phosphoglucolactone is broken down to 6-phosphogluconate. And the enzyme that's going to break down 6-phosphoglucolactone will be called
G6PG lactonate.
Yeah, it'd be like 6-phosphoglucolactonase. All right. Now we're again making another NADPH, right? Which means that we took a hydrogen from this and we added it to NADPH. What's the name of the enzyme that's going to do that, Cole?
Another dehydrogenase.
Yeah, it's another dehydrogenase, right? This one's going to be called 6-phosphogluconate dehydrogenase. And that enzyme ultimately produces ribulose 5-phosphate. All right? Ribulose 5-phosphate is the very first step of the non-oxidative phase. So that concludes the oxidative phase. We're getting the oxidative phase called that because we end up producing these NADPHs. We also, in this very last step, produce CO2. I don't think that's particularly important for you to know for the MCAT either, but just information for you there. Okay, so we, in the non-oxidative phase, start with ribulose 5-phosphate. And ribulose 5-phosphate will be converted into ribose 5-phosphate, which is what we want, right? As the ultimate product of our pentose phosphate pathway. So what's going to convert ribulose to ribose? If they're, I'll give you a hint. They're made up of all of the same atoms. They're actually isomers of one another. So what's going to convert ribulose to ribose?
Isomerase.
It's an isomerase. That's exactly it. And what's the name of that isomerase going to be if
Ribulose 5-phosphate isomerase.
Yeah, if biochemists are particularly creative people, then it's going to be called ribulose 5-phosphate isomerase. Okay. There's also an epimerase that's going to create xylulose 5-phosphate. Epimerase. And that creates xylulose 5-phosphate. Okay. And then the two of these together. Okay, so first of all, we can stop here and take a break, right? We started with glucose 6-phosphate, which we got from what? Where did we get glucose 6-phosphate?
From glucose.
Right, we got it from glucose. From what biochemical pathway?
Glycolysis.
From glycolysis, right? So we got glucose 6-phosphate from glycolysis, and we use it to eventually make ribose 5-phosphate, which you need for all of our ATPs and like DNTPs, DNA, RNA, all of that stuff. Okay. When we don't need ribose 5-phosphate anymore, then we can shuttle all of this stuff back, all of these carbons that they have energy, right? We can shuttle all those carbons back into glycolysis, all right? And we do that by converting ribose 5-phosphate and xylulose 5-phosphate into, to fructose 6-phosphate and to 3-glyceraldehyde phosphate, glyceraldehyde 3-phosphate. Okay. So glyceraldehyde 3-phosphate and fructose 6-phosphate are both parts of what biochemical pathway again?
Pentose.
It's
Glycolysis?
glycolysis, yeah, exactly, right? So we got our carbons, glucose 6-phosphate, from glycolysis. Ultimately, we return them, if we don't need ribose 5-phosphate, we can return them to glycolysis, all right? And the enzymes that make that happen are transketolases and transaldolases. And that's particularly all you need to know, all right? So we can take xylulose 5-phosphate and ribose 5-phosphate. We can convert them back into, the first thing we can get is is glyceraldehyde 3-phosphate. All right, we can convert. There's going to be something else that gets produced here, and that's not particularly important for you to know. But these two things, again, we can use another enzyme to produce fructose 6-phosphate, and then we can use another enzyme. There's going to be two things again here produced. We can use another enzyme to produce fructose 6-phosphate and glyceraldehyde 3-phosphate. All right. So the enzymes that make that happen are transketolases and transaldolases. Transketolase the transketolase is going to convert xylulose 5-phosphate, ribose 5-phosphate to. I frankly I just don't even want to. I don't want you guys memorizing this for the MCAT. It's called cetohepta 7-phosphate. It's not particularly important, but it's there for you in case you need it. And then a transaldolase is going to convert these two. Transaldolase is going to convert these into, again, something I just don't even want you to know. It's just another sugar backbone, right? It's got 4 carbons this time. And then another transketolase to create glyceraldehyde, 3-phosphate, and fructose 6-phosphate. All right. The final thing I want you to know is the rate-limiting enzyme for this entire reaction is, well, what is it going to be? What do we start with? We started with what?
Glucose 6-phosphate.
Yeah, we started with glucose 6-phosphate, and where did we get that from?
Glycolysis.
Glycolysis, right? So if we hadn't gotten it from glycolysis, then, well, we would use glucose 6-phosphate for glycolysis. The rate-limiting enzyme for this entire pathway is going to be the conversion of glucose 6-phosphate to 6-phosphoglucolactone, all right? So it's glucose 6-phosphate dehydrogenase. That's our rate-limiting enzyme. And it's allosterically stimulated. So this is, first of all, rate-limiting. And it's stimulated by the presence of NADP+. It's inhibited by the presence of acetyl-CoA. All right? And that is the triple P pathway.